My name's Joe Zock. I'm the Senior Director of Product Management for IntelliCyt, a Sartorius company, and welcome to SLAS 2017. Today we're going to talk about the iQue Screener Plus family of products for suspension cell and bead screening. We have two new configurations of the iQue Screener Plus. We have now a VYB, a violet, yellow, blue laser configuration, which is really good for fluorescent proteins and multiplexing fluorescent proteins. And we have a BR, or blue-red configuration, which has 6 channels, which is really good for antibody screening and downstream bioprocessing. The iQue Screener is a tool for doing suspension, cell, and bead screening. It's extremely fast. We can process 96-well plates in 5 minutes, 384-well plates in 15 minutes. We even have a version that can do a 1536-well plate. It is also very miniaturized. We can take as little as 1 microliter out of the well and process it through the detector, getting all the data that you would get from a standard flow cytometer without the dead volume and with the speed of 35,000 cells a second. The system is completely automated. The plate comes in, it goes into the system, and then all the washing and cleaning between wells, in the middle of a plate, after a plate, can all be done automatically with these fluidics that are on the front here. The goal is to process thousands and thousands of samples. And that is a need in immune target screening in big pharma. It's a need in antibody drug discovery, where you're looking at antibody libraries and you're trying to find what's interesting. And it's also a very big hit in immunotherapy, where people are engineering CAR T cells and they're trying to discover, are the CAR T cells they've made killing the cancer cells? We can do that in the dish in a 384-well format in under 20 minutes. The iQue Screener Plus is completely automatable, and it's also has what we call the QMax refill station. So this accessory gives you the ability to extend your capacity to screen from 7 hours runtime to over 50 hours runtimes. And because of the hot swap capability, you can never, ever have to stop your experiment because you need to change the fluidics. You can do it on your time. People always ask, what makes this different? Why is this not just a flow cytometer? And there are 5 major reasons. We call them the IntelliCyt advantage. Number 1 is speed. We can literally process plates in minutes. The second advantage is miniaturization. This means that the volume of reagents that you have to use to get the same answer can drop dramatically. And that means that running these screens is now more cost effective than it ever has been. The third one is usability. Everyone loves usability. That's why it's the heart. This system is so usable to set up. Once you set it up, it's templated, so the next plate that comes through gets exactly the same analysis, and what you end up with is a way to get to actionable data very, very quickly. The fourth one is content. If you're doing immune screening, immune target screening, antibody therapy screening, you're looking at checkpoint inhibitors, you're looking at CAR T cells, you need to know from an immune standpoint what's going on in that well, and we deliver that by bringing bead assays that can capture cytokines, along with the cells themselves, their health, their population dynamics. We can immunophenotype every immune cell population that's in there and keep track of them all. And we can do that all together in one well at one time in a 384-well plate in under 20 minutes. That's what content is. We're the most high content immune system assessor in the industry. The last one, which is really, really important, is InSight, right? It's actually our title, InSight at the speed of light. What InSight is the ability to get an actionable piece of data, something that you can use in the shortest amount of time possible. We have the ability with our software, with profile mapping, to take the huge amount of data I just talked about and boil it down to the 3 wells that you might need to look at in that plate. And we can do it automatically and instantaneously. The iQue Screener Plus platform is an integrated solution of instrumentation, multi-site reagents, and 4-site software that provides a complete solution for immunophenotyping and immune targets, for antibody screening, and for cell therapy applications like CAR T. So a new addition to the IntelliCyt family is viral analysis. And this is the Virus Counter 3100. It has the ability to look at viral particles themselves in a biologically relevant way by using a combination of a protein dye and a nucleic acid dye or a set of antibodies to bind specifically to the virus in question, we can now characterize and count actual virus particles. This is a significant advance for understanding virus particles in bioprocessing and vaccine developments. So in conclusion, for suspension cell and bead assays, the kind of assays that the antibody screening community needs, the kind of assays that will allow you to do immune targets like checkpoint inhibitors, immunotherapy work where they're taking the cells out of a patient, they're genetically modifying them, we're testing them, and then we put them back in the patient. This is unprecedented in medical science. They've never seen the kind of clinical results that they're seeing. We are the best at it, and we'd love for you to see more. So please come to www.IntelliCyt.com and find out more.